Good morning, everyone. So hello, I'm Yui Qing. I'm a postdoc at Schmidt Center, and I have the honor today to introduce you to Emma Lundberg. Emma, welcome. So Emma has been a professor at KDH Royal Institute in Sweden, and where she created and served as director of the Cell Atlas part of the Human Protein Atlas. And the Cell Atlas images, you already seen a lot actually yesterday through the talks of the speakers. So Emma moved to Stanford a few years ago and is now also serving as associate professor at bioengineering and pathology department. So where she is really using deep learning to utilize all the data we've generated in the proteomics and imaging to advance the modeling and understanding of spatial temporal organization at cellular and subcellular level. I personally had the honor of collaborating with Emma like a few years ago. Best experience so far, and even the 8 a.m. meetings are so enjoyable, I have to say. And kind of like Emma has been pioneering on the science side, but also like the way we actually do science. Emma need to put a spoiler, but she has hosted 2 Kaggle challenges, and also, 3, I'm sorry, correction, 3 Kaggle challenges, and also like most amazing, like collaborating with video gaming company and really allowing every single person to be able to contribute to science. Well, I can go on and on telling you how amazing like Emma is. And before I put out more spoiler alert, Emma, please take away.
Thank you. That was a very sweet introduction. Thank you very much. She's amazing, by the way. I've enjoyed working with her. So today I'm going to talk about, well, what we touched it many times yesterday. So how we can use spatial proteomics to do cell modeling. And we've been talking about virtual cells. And can we use AI to model cells, for example? And I think this is a very interesting field. And I do think that at some point we're going to be able to do it. And I've had the pleasure of working with the Chan Zuckerberg Initiative last year. And we organized a series of workshops and really discussed this topic more deeply, which ended up in a perspective paper where we discussed if we build virtual cells, what should they be able to do? What's needed from the community? How would you possibly build these cells and such? But I just want to point out that a key challenge will be to connect the scales, as we all know and as we've discussed already, from molecules to cells, from cells to multicellular system. And this is not trivial. And I will focus on how we can do this at the subcellular level in my talk today. And I would argue that protein data will be key for AI virtual cells because proteins are the building blocks of human biology. They execute most of the functions that we have in cells. Transcripts do not. And cells are 3D systems consisting of nested functional units, and we really want to understand this structure. And we also know that there's a lot of moonlighting activity or pleiotropic effects that arises at the proteome level that is very hard to predict from the sequence level. I would also argue that images will be key for AI virtual cells because they're so rich in information related to structure and morphology. They can capture both spatial and temporal scales, again, trying to meet that critical gap of how do we bridge the scales in virtual cells. And I also think it's very cool to think of microscopes as opportunities for developing autonomous instrumentation and very tight systems for lab-in-the-loop experiments. So with that little backdrop, I'm going to get to my talk for today. So at first, I will talk a bit about the work that we've done to map where proteins are in our cells, and then I'm going to give you some different vignettes of how we're trying to model cells off from this data. So the interest of my lab is really to measure and model the spatiotemporal subcellular organization of the human proteome with a goal to understand how variations in protein distribution can fine-tune cellular function and also how mislocalization can lead to disease. And we've been doing this type of work for a long time, and we're very proud producers of the Human Protein Atlas project, where I'm a co-director. So this project it's been going on for 20 years now, and it's an open project, and we've generated antibodies for the majority of all human proteins that we can deploy in a variety of assays to map where proteins are expressed. If you haven't checked it out, you can find it at proteinatlas.org. It's fully free, no registration needed. And it's actually, we're gaining more and more traction. So the number of users increase every year. Right now, it's about 2 million users per year. And I should say we have nowadays one update every year. So the next update with new data is coming in October this year. And the key data in the protein atlas is images. We have about 20 million images that either are very classical, like no fancy imaging right now. Immunohistochemistry to the left that shows protein expression in cell types of most tissues of the human body, or confocal microscope images that shows the subcellular expression of proteins within cultivated cell lines. And today we do work with both of these image data sets, but today I'm going to focus on the subcellular aspect of this image collection. So the images have been generated over many years, very systematically, with some reference markers. So we have nuclear staining, a staining for microtubules, a staining for ER. So this makes it possible for us to kind of orient ourselves in the cells. And on top of this, we've used our in-house generated antibodies to localize most human proteins one by one. So CD44 would look like this, and let's see if the pointer works. Yeah, we can see that it's expressed at the cell membrane, plasma membrane of this cell. If we see an image like this, we'll say that this protein in green is localized to actin filaments. Everything has been manually annotated over the years. This protein, we see these two dots here, that's the centrosome, and so on. So we've done this for a long time. When we published the first paper, the subcellular map of the human proteome, we actually provided the first evidence for localization of that protein for over half of all the human proteins. So it's very basic information, but still very valuable if you want to understand protein function. And I'm not going to talk about that today, but I do want to say that in the last release, we added some new data that we've been working on for many years, where we've actually localized proteins to the primary cilium, which is a very interesting cellular structure that is involved in a lot of different diseases and also been linked to diseases like obesity and autism. But that's for a different talk, but happy to chat about it in the break later if someone is interested. So some of the key findings that still fascinates us is that here you can see kind of the classical organelles. This is a protein in the Golgi apparatus, in the mitochondria, and so on. But we find a lot of proteins that are in multiple compartments, as you can see to the left here. This protein is on the cell surface and in the nucleus. And most proteins execute their function by interacting with other molecules. And it is likely to assume that the potential interactors in the nucleus and the plasma membrane compartments are different. So it might be so that this protein is performing two different functions in these two different compartments. The other fascinating aspect is, of course, when we see variability, single cell variability. This protein is expressed at low level in this cell, at a high level here. It's actually a cell cycle protein. So this kind of hint at some kind of temporal dynamics. So even though the images are static, we can still assume that there is sometimes see signs of temporal dynamics. Most often it doesn't look this clean. Most often it looks something like this. So this is actually a very well-studied protein. It's enolase 1, and we know that it has different functions in different compartments. So these are all, it's a cell line, all genetic, kind of even transcriptionally very similar cells. But if we look at this protein, they're very different. Enolase 1 performs its glycolytic canonical function in the cytosol. It controls gene expression in the nucleus. And on the plasma membrane that you can see here, it's actually functioning as a plasminogen receptor. So these cells, even though they're genetically and transcriptionally highly similar, present very different functional capacities just by changing the amount and the location of this one protein. So we're not interested in enolase 1, we're interested in this at a systems level. So how common, if we look at the entire proteome, how often do we see this? So we've been able to show that between 50 and 60% of all human proteins localize to multiple compartments. And I dare to say that I do believe that a lot of these proteins perform dual functions, and there's a lot of unknown biology in there. And about 20% of all proteins show signs of temporal dynamics. And I think this number is still an underestimate, so probably it'll get higher. So with these conclusions, I would want to argue that I don't think that we can build good models of cells unless we take this spatial proteome information into account, because otherwise we're missing out on so much biology, basically. So how can we do that? So I want to present 3 different vignettes. And the first one is just, can we make use of a very simple metric, which is cell shape, to try to understand all of this? Because we have a great image collection, but it's one protein at a time. There's still no technology so that we can multiplex 10,000 proteins and visualize that in a single cell. So can we use different computational frameworks to try to aggregate the data, basically? So one is to try to use shape modes, and this has actually been a very fun collaboration with Susan Rafalski sitting there and Matthias Viana at the Allen Institute. So we've constructed an interpretable shape space to study the human proteome, and we've then mapped all of these proteins that we've measured onto this shape space. And these are some of the cell lines that we've been working with. So I just want to show that they are, of course, they look very different. We have more epithelial or fibroblast-like cell lines, and there's also some blood hematopoietic cell lines and so. So we used the same framework that Susan and her group developed and showed in her, I think she briefly showed that Nature paper on her slides. So basically, we start by segmenting the cell in the nucleus, and then we basically trace the contour here, and you get this wave-like pattern, and then we perform a Fourier transform so that we get these coefficients. And then we do dimensionality reduction, and then we reconstruct the cell. So we get the average cell shape. And then we can walk along, do a standard deviation walk along the PCs to see what the shape mode variations are. So if we do this, in these plots you can see how the cell shapes change in the different, this is PC1, PC2, PC3, etc. And these are, of course, they don't look like the real cells, but it's basically this is basically based on the assumption that cells have very complex shapes, but maybe they're made up of a series of common shape mode variations that we can combine. So for example, here you can see all the different cell lines that we tested. You see the average cells look pretty similar in shape, but different in size, slightly different. What is very cool, though, is that the shape modes are highly conserved and basically exactly the same for all of these cell types, even though they're different. So we have like the size length changing, the size and the polarization changing, the nuclear positioning, the width versus length, and kind of center of mass shifts and things like that. So now when we have these shape modes, can we then map protein data onto it, and does it make sense? So we've tried doing this for comparing cell shapes, organelle shapes, pathways, single proteins, and I will take you through some of that. So first of all, when we mapped the protein expression, we ended up using deformation fields. So basically we deform the real cell staining into this average cell shape. And then we can do that for, let's say, pick 200 nuclear proteins, and we average them all, and then we get this average nucleoplasm staining in a given cell shape. Same thing for the mitochondria. So it becomes, of course, very diffuse because it's the average, but we still see different patterns for all the different organelles. So what we can do then with these different patterns is that we can correlate them and see how do the different organelle distributions correlate in different shape modes. So thinking of what Jennifer talked about yesterday, she showed beautifully with her cryo data that the correlation between organelles is highly conserved, and we're actually seeing the same thing. So this is in one of the cell lines, the U2OS cells. We see this is the correlation between all the different organelles, and of course it makes biological sense, like the nucleoli correlates to the nucleus and so on. But what is very cool is that when we compare the extreme, and this correlation looks slightly different for different cell types. But when we compare this correlation in the standard cell shape and the extreme versions, we compare the round cell to the elongated cell, for example, or we can see that this correlation is highly conserved across all these shape modes, even though it's slightly different for the different cell types. So that's good. That means that we should be able to, like, this structure is preserved, and it means that maybe there's some kind of generalizability lying in here when we think about virtual cells too. And then we thought about what if we try to look for variable proteins and see if we can find certain proteins or pathway that are enriched in certain shape modes. So we did this, and here you can see how different proteins are expressed in the different PCs, the different shape modes. And we found over 500 proteins that vary significantly through shape space. A lot of them are cell cycle proteins, of course, but many, and the enriched pathways are interestingly related to both cell cycle, metabolic, and signaling processes. And we can go even further and start to look into really like fine-grained things. So as an example, I'm just showing, we had a cell cycle marker in a big image data set. So we said, let's look at cells in one cell state. Let's look only at G2 cells. And then we compare round cells versus elongated cells in G2. Do they show any difference in protein expression? So when we aggregate this data of differentially expressed proteins here, we see that we see different pathways expressed. And it seems like the round cells are really focused on oxidative phosphorylation and seem to be preparing for division, whereas the elongated cells show enrichment for pathways that relate to lipid metabolism and migratory patterns. So maybe they are actually preparing to migrate and not directly divide. And of course, we can go back to the proteins that kind of contributed to this enrichment and see that these are the cells that we can classify as round cells and elongated cells. And yes, we can actually visually also observe this difference in expression. But it is a lot easier to interpret in this aggregated shape space. So in total, in summary, we believe that these interpretable shape space are it's a very useful pipeline, maybe not the right tool to build a virtual cell model, but I think it's a very good pipeline to understand your image data sets. Let's say if you do an optical pool screen and you want to see how do my population of cells change, for example. It's a great pipeline to just understand the baseline of cell distributions that you have and if you have any significant shifts in this space. I didn't talk about it here, but we can also do this at the protein level upon drug perturbation, and then it turns out that it's very helpful to stratify the data into shape modes before you start to look for significantly affected proteins as well. So with that, can we, besides aggregating the data like this, can we think about building spatial cell models, and what could that be? What's the best spatial model we have right now? Probably gene ontology cell component, still some kind of hierarchical cell structure. So can we use our data to build something like that? And we've worked recently, in collaboration also with the Chan Zuckerberg Initiative, to build Subcell. It's a suite of self-supervised deep learning models for fluorescent microscopy. And what we wanted to do here is to build a model that can both classify patterns, embed spatial patterns of proteins, but also understand morphology of cells and phenotypes, because I think those are the two main use cases, like drug perturbations, CRISPR perturbations, and also localization of proteins. And of course, we want to have a model that can provide us insights to biology beyond what we can kind of perceive with our eyes. And the hypothesis here is that we can, while our data set is not the largest in terms of number of images, like the JUMP data set is big, there are some massive data sets out there, but it is the most diverse data set when it comes to patterns, because we don't only have 5 markers, we have all human proteins. So we thought that even if this is a smaller data set, it's still a pretty big data set. We can build a broadly useful microscopy vision model, and hopefully this model will have learned to pay biologically relevant attention because we've trained it with images of all intracellular human proteins, more or less. So this is the architecture that we ended up with. It's a vision transformer, basically, with 3 different losses and tasks. So it's a reconstruction task, which is usually great for building models to classify morphology. It's not great. Those models aren't very performant when it comes to classifying subcellular patterns, though. So that's why we added also 2 different contrastive functions here, one focused on the cells and augmenting it, and one focused on different proteins and making assumptions that this protein is more likely to have a similar pattern in a different cell line than compared to a completely different protein, for example. So we're making some assumptions about biology here. And I will show you the results for our top 2 best models. So we first of all test them on the 2 tasks that we want to do: pattern classification and then phenotype classification. And here we just use, can we classify different cell types, a very simple task. And we compare it to what we consider to be state-of-the-art models, the Dino for Cells model, and then also what we call best fitting. So this is the winning model from one of our previous Kaggle competition, which is the supervised model. And basically, our models outperform the state-of-the-art in both of these tasks, so that's a very good starting point. But we also want to go beyond what the human eye can do. So in order to test this, we try to predict pathways from these embeddings and also predict protein-protein interactions. And of course, it's performing better than the other models, but it's very hard to predict pathways only from microscope images, but we can actually start to predict protein-protein interactions pretty well, and also outperforms the other models here. We then started to analyze the attention to kind of go back to our hypothesis. Was it useful to train on the protein atlas data? And we were so surprised when we saw the patch attention generated by, in the transformer. So these are all the different attention heads. And if you look at these images, it almost looks like immunostaining or different organelles. Attention head 4 clearly only pays attention to the nucleoli. Attention head 7 looks at this region on one side of the nucleus. Attention head 6 has kind of more membranous, general, all membranes in the cell type of attention. And let's see, attention head 12 looks at the plasma membrane. So it's kind of cool that the model has taught itself to look at regions in the cell where we know that proteins are distributed. And we believe that these are biologically relevant regions, and that's maybe why the model has good performance. And if we also look at the correlation of the attention to the different tasks that we're trying to do, and these are just a lot of different cells where every dot here is a cell, and then we just link the attention to the different cell lines and also the different protein locations, we can see that these are really encoded orthogonally here, right? So the model has learned to think about both morphology and location of proteins without confounding them too much, which means we were very excited about this because this means that we can start to maybe compare protein distribution patterns in cells that have very different morphology, which has been very hard to do before. Besides that, we wanted to test this model on other data sets. No fine-tuning here. We just tested exactly as is. So we tested it on the open cell data set, which is a data set with just fewer markers, 2 markers. And it outperformed the model that was specifically trained on open cell. We also tested it on a bridge to AI data set that we've generated, but this is a new cell type that it hasn't seen in training, and it's also drug-perturbed cells. So we were able to show that we can bring all of these drug-perturbed cells and other cells into a joint embedding with this model, and we can identify relocalizing proteins upon drug perturbation in this embedding space. And then what we were most excited about is that when we even apply it to the JUMP data set, which is images of quite different resolution and such, we can actually outperform the existing models that were trained on JUMP in tasks like mechanism of action prediction. So again, we believe that this is because the model has learned to pay attention to biologically relevant regions. And of course, we're excited to start to fine-tune the model for some of these tasks as well. But with that in mind, we're pretty confident that this is a robust model for embedding cells from microscope images. So what can we do with that? Can we embed all the images from the protein atlas and start to build some kind of graph that looks a bit like the gene ontology cellular component? And yes, we can. This is the graph that we get if we build it from all the human protein atlas images. You'll have a hard time to read the labels here, but this is the cell. And then we do just hierarchical clustering, and you can see that, and the labels here are from either Go cellular component, at the bottom layer it's from Go biological process. You see we get the nucleus, the cytoplasm, and then that splits up into like the cytosol, the endomembrane system, and filaments, and deeper and deeper. So basically it recapitulates what we know about biology and how the cell is organized. But then when we start to get down to this level, we're seeing clusters that we couldn't have annotated by eye, and this is where it's interesting to start to inspect them. So for example, here we see a cluster of proteins involved in translation, ribosomal proteins. We could not have annotated that. I also really like these clusters. So it's a sub-split of of proteins localized to the nuclear membrane, and when we look at the proteins in these different classes, one of them is structural proteins that are involved in maintaining the structure of the nucleus. One of them is protein involved in export from the nucleus. One is protein involved in import to the nucleus. So it's very cool that we're starting to see these functional differences that we could never, ever have annotated by eyes. I do think that these models capture information that is relevant to understand biology. And this model, as a collaboration with CCI, it's available on their virtual cell modeling webpage, and there's also a demo where we use this model to understand how SARS-CoV-2 relocalizes protein upon infection. But with that, even if it's nice that we can have this graph of cells, how can we go beyond? Like, there's a limit to the resolution in our images. It's confocal microscopy. How can we try to go beyond that down to kind of protein-protein interactions? And how can we improve the spatial precision of these cell maps to true complex level? And this is the project that we've been working with UA on. So it's been basically 2 publications. And we had the most recent one now in Nature 2 weeks ago. So this is from the newer paper. So the idea here is that maybe we can build such an hierarchical map if we integrate 2 types of spatial data. The image data that describes the global distribution of proteins in a cell, and the protein-protein interaction data that describes the local distribution of proteins in the neighborhood of any given protein. And we can use machine learning to integrate those data types and build a hierarchical model of the cell. So for this, we used the UTOS cells and all our images we've generated, and then Steve Giggy and Ed Hutlin here at Harvard generated protein-protein interaction data. We're still limited for data. So this is not a proteome-wide map. This is only 5,000 proteins, and we're limited because of the lack of PPI data. So if anyone is interested in generating protein-protein interaction data at scale, that would be incredible. So the first thing that we do is, of course, that we embed each data type alone with our machine learning models and a node-to-vector graph model, and then we bring these embeddings into a joint embedding space. And then here's the joint embedding space. We can, of course, start to look at it and see that, yes, we're keeping all of these kind of clusters that resemble organelles. And if we go further in, we can also, again, start to see these clusters that are more biologically refined, probably driven by the protein-protein interaction data. So there's definitely biologically relevant information in the structure of this embedding. And what we also do then is that we use this trick that actually works pretty well, that we calibrate the size of the clusters to basically, this is a bit complicated to describe. Basically, it turns out that the size of any structure in this assembly correlates very well to the number of known proteins in that structure. So the size of the Golgi apparatus, according to literature and structural studies, corresponds to the number of known proteins in it. We get a pretty nice linear correlation there. So we use this linear correlation in order to be able to estimate the actual size of systems that we can identify in this embedding. And in this embedding, we can do hierarchical clustering and build a similar map to the one that I showed you before. There are some differences here. Proteins are allowed to be in multiple nodes, for example, if they perform multiple functions. And as you can see, we have all these major organelles, but then we have a lot of the smaller kind of systems here driven by the protein-protein interaction data. In total, we identified, this is nearly 300 different cellular systems. All the yellow ones are known in gene ontology and literature. All the purple ones are new. We've spent a lot of time validating that they are indeed real systems, but that was more described in the first paper, so I'm not going to go through that now. Instead, I'm going to focus on how we can use these maps and to do better functional genomics and better understand biology. So, for example, here is an interesting system, the RAG regulator complex. It's known, we have a structure for it, but we found 2 additional proteins in this system. Okay, how can we validate that? So here we worked with Andrei Shali at UCSF to do structural modeling, and he showed that these 2 additional proteins, BORC-S6 and ITPA, can actually bind to the known RAG regulator complex in this way. So basically, these systems can be used to guide AlphaFold and kind of structural modeling principles, and it would be kind of cool to think about folding everything with everything using these maps as a kind of scaffold to reduce the search space. So that's one way you can use this map. Another way is that we use this map to use as assemblies, as convergence points for pediatric cancer mutations. So here we took very big pediatric mutational data sets that have been published, and when we map it onto these assemblies, we identify that there are certain systems that are consistently disrupted in certain pediatric cancers that were not discovered in these initial papers because the mutations might be hitting different genes, but for example, these genes are often involved in collagen biosynthesis or in this nuclear complex associated pores. It's also a way of using, just aggregating data on top of it, a data-driven cell map, basically. What we also did, which I think is interesting, we were able to propose function for 46 proteins that have no known function, and we were also able to suggest additional secondary functions to 951 proteins using a guilt by association principle by just these assemblies. So this map is currently being uploaded into the EBI complex portal, and we hope that people will use it. I personally think it would be very nice to see more data-driven cell maps like this that have not been biased by our assumption of biology and how biology should be. And I think it would be very interesting to have maps for different cell types as well, so we can understand how much rewiring there is in a cell between different cell types. So with that end, a very, very, very short ending. I also think that this is cool, but the image space is even cooler. So I kind of want to pull it back into the image space again, because again, it's interpretable, it's close to the bench, and it's a place where we can do a lot of validation experiments. And this is where we think about generative models as kind of virtual microscopes. So at some point, we also want to go towards that lab in the loop where we, well, predict where a protein would be, or a pathway, which pathway is affected, where would this protein be, which experiment can we do to validate it, and then do that experiment in the microscope. And then we want to generate synthetic images to do so. So we've trained models that can generate very realistic images de novo. We've trained organelle-specific models so inputting these different organelles, we can generate images of the other organelles, for example. And we're also training a model with the aim to basically build a synthetic image that multiplexes all human proteins so that we can bring these maps back into the image space. But for many, many reasons, this is a difficult task. And I don't think I have time to talk about that now. And of course, in the end, ultimately going from sequence to image would be a goal, but there's a lot of problems to solve before we're there, and we probably need a lot of additional data types to do that well. And with that, I'd like to thank my group, and thank you, and I'm happy to take any questions.
Hi, very nice talk. I was wondering for the protein interaction prediction, can you actually predict like real interactions versus co-localization into the same compartment?
Yeah, so what we did in that plot is that of course, what we can predict in the image space is probably co-localization, but that has a pretty good correlation to interaction. So when we compare it with it, we try to predict the string score, which is a likelihood of interactions. So we can predict the string score pretty well. But of course, it's not going to be, we can't do anything beyond the resolution of the images.
Oh, just a follow-up question. Have you tried to look into like co-evolution of those proteins that you predict to be interacting together? If they co-localize and they interact, you might see co-evolution signals.
Sorry, can you?
Like, have you tried to look into co-evolution signals of those proteins that you predict to interact, since they are localized at the same place and interacting, and maybe with, multiple sequence alignment or AlphaFold, you can explore that a bit?
Yeah, that's a very good question. So what we've done a lot of things there, but then we realized that there's a lot of papers that just predict locations from sequence, mitochondria. It's described as a solved problem in literature, and it's actually not solved. I didn't present that here. So we've spent a lot of time making a better benchmark data set than what the community has used that is highly validated, not only Protein Atlas data, but other like two sources of experimental validation. And when we applied that benchmark data set is also balanced with all locations and balanced in terms of sequence features. And when we apply the best models today on predicting location on that benchmark, they all perform very well. They perform pretty okay on nuclear, cytoplasm, and some other locations, but certain cellular compartments, they can just never predict. So I think that this is where we need more information than amino acid sequence alone. We probably need regulatory regions, UTR regions. We have also been trying different protein embedding models. Some of them work well if it's like a long intrinsic disorder region. It might work well in predicting some locations, but certain times it's just one amino acid that is changing, and most models don't capture that. So I think that it's nothing that my lab will do, but I think it will be very interesting to start to think about location as one of the features you want to predict with protein models. And when you design molecules and new proteins, maybe not only design the protein with a certain function in mind, but also with a certain location in mind, because there are certain classes of proteins where that location really matters for its function.
Awesome.
It's not really an answer to your question. But
So when you look at your new ontologies that you're finding, do you also find places where you want to, or your model or your data wants to change the tree structure? Because over the years, predicting protein function, a lot of people who do that work complain that that tree is not always right, I guess.
Yeah, I think that the high level tree, like the organelles, are pretty robust and right. Yeah. Down there, I think there's a lot of rewriting to do, and I also think that we're still a bit data limited, because when it comes to like below the mesoscale, where you have a lot of phase-separated systems and things like that, we're probably going to have to build sequence image models to really capture that, and I'm guessing that we're going to see a lot of shuffling in these ontologies when we do that.